Hello everyone. Today we are going to discuss about the physiology of blood clotting. What is blood clotting? Blood clotting, also known as hemostasis, is a complex process that stops bleeding by forming a blood clot at the site of injury. This process is essential for preventing excessive blood loss and maintaining tissue integrity. Without proper blood clotting, even minor injuries could lead to severe complications. And mechanism of clotting. Simplest scheme of clotting was proposed in 1903 by Morwich. When blood is shed, the platelets, also known as thromboplast, disintegrate and liberate thromboplastin. Some amount of thromboplastin is also derived from the damaged tissue. Thromboplastin then convert prothrombin into active thrombin with the help of calcium ions. Then the thrombin interact with fibrinogen forming fibrin, which is the clot. Thromboplastin plus thrombin plus calcium ions is converted into thrombin. Thrombin plus fibrinogen is converted into fibrin. Next, we are going to see about cascade theory. The enzyme cascade theory explained the coagulation of blood. This theory was proposed by Biggs and Macfarlane in 1966. Enzyme cascade refers to a series of enzyme catalyzed reactions, each reaction initiating the next until the final step is reached. According to this theory, clotting in a wound occurs in four steps, namely formation of prothrombin activator, formation of prothrombin, conversion of prothrombin into thrombin, conversion of fibrinogen into fibrin. Next, we are going to see about flow chart of blood clotting. There are 7 steps. First, injury, the second prothrombin activator, the third prothrombin, fourth thrombin, fifth fibrinogen, and sixth fibrin, and last one seventh is clot. Next, clotting factors. 13 factors are there. Factor 1, fibrinogen, a soluble plasma protein. Factor 2, prothrombin, the precursor of thrombin formed in the liver. Its synthesis depends upon the presence of vitamin K. Factor 3, plasma thromboplastin factor, a protein found in plasma and tissue extract. This converts prothrombin into thrombin in the presence of several coagulation factors and phospholipids. In case phospholipids are absent, these are provided by blood platelets in the form of cephalin. Factor 4, calcium. Calcium ions. Factor 5, labile factors found in plasma but consumed during clotting and not present in serum. Factor 6, no such factor. Factor 7, stable factor or autoprothrombin found in both and plasma and serum. Factor 8, anti-hemophilic factor, a macromolecular complex composed of AHF and one Wilbrand factor. The latter accounts for 99% of mass of factor 8, which is glycoprotein synthesis by megacytosis and is consumed during clotting and absent in serum. Factor 9, Christmas factor, found in plasma, it activated during clotting, hence its activity in serum is much greater than in plasma. Factor 10, Stewart power factor, present in plasma and serum. Factor 11, plasma thromboplastin antecedent, found in plasma and serum. Factor 12, Heachman factor. This factor is found in plasma. It was found to be absent in Heachman family, hence the name. Factor 13, fibrin stabilizing factor, found in plasma, which converts soluble fibrin into insoluble fibrin. Steps involved in blood clotting. Clotting occurs on the injury. During the injury, the blood vessels is ruptured. The endothelium is damaged. The collagen lying beneath the endothelium is exposed. Factor 12, Heachman factors, comes in contact with collagen. Factor 12 is converted into activator factor 12 in the presence of kallikrein and kinogen. The activator factor 12 converted factor 11 into activator factor 11 in the presence of kinogen. Activator factor 11 activates factor 9 in the presence of factor calcium. Activator factor 9 activates the factor 10 in the presence of factor 8 and calcium. When platelets comes in contact with collagen of damaged blood vessels, it gets activated and released phospholipids. Now the activated factor 10 react with phospholipids and factor 5 to form prothrombin activator in the presence of calcium ions. Prothrombin activator is also produced in the another route. The damaged tissue of the injury release the factor 3. Thromboplastin contains protein, phospholipids, and glycoprotein, which act as the proteolytic enzyme. Glycoprotein and phospholipids components of thromboplastin convert factor 10 into activated factor 10 in the presence of factor 7. Activated factor 10 react with factor 5 and thromboplastin to the form prothrombin activator in the presence of calcium ions. Prothrombin activator converts prothrombin into thrombin in the presence of calcium. The thrombin itself initiates the formation of more and more thrombin. The initially formed prothrombin activates factor 5. The factor 5 accelerates the formation of prothrombin activator. The prothrombin activator converts prothrombin into thrombin. In this way, more and more thrombin molecules are produced to accelerate clotting. Thrombin converts inactive fibrinogen into active fibrinogen. The active fibrinogen is called fibrin monomer. Fibrin monomer polymerize with one other to form loosely arranged strand of fibrin. The loose strand of fibrin are modified into tense and tight fibrin threads by factor 13 in the presence of calcium ions. All the tight fibrin threads are aggregated to form a meshwork of stable clot.